Hello everybody, welcome. So today we're going to talk about total yeast and mold. If you are in the lab industry, you're probably very familiar with total yeast and mold. If you're not, some of this may be very new to you, so I am going to do a brief overview at the beginning. So what is total yeast and mold? So when we talk about total yeast and mold testing, we're talking about looking at quantitatively the number of colony-forming units that are present in 1 gram of sample, or CFU per gram. That's how we quantify it. When we report out a result, we say there's 3,000 CFU per gram, or 10,000 CFU per gram, or whatever that number may be. And total yeast and mold can include so many different species. It includes everything. Every total mold or yeast you can think of, it includes. So Aspergillus, penicillin, Candida, Sportabolus, Pichia, Fusarium, and many more. And that's one of the complexities with doing total yeast and mold testing. When you're looking for so many different things, how do you know if you're seeing them all? And how do you know if everything you're seeing is dangerous or if everything you're seeing is good? What is the problem that arises with total yeast and mold testing? So total yeast and mold testing in a laboratory, there's 3 primary ways that laboratories are providing total yeast and mold testing. The first being culture-based plating methods, and that's going to be using an agar or a petri film or a membrane filter and growing colonies. They're going to take a sample, dilute it with a media, add, the sample to the plate, put that in the incubator, and then wait for it to grow and see what happens, and then they're going to count those colonies. The next way is going to be qPCR methods, or quantitative polymerase chain reaction methods. And what that's going to do is you're going to take the sample and you're going to completely isolate the DNA. You're going to isolate both the cannabis DNA and the microbe DNA. After that, you're going to use primers and mix that with that, the primers that'll look for both the cannabis DNA and the microbe DNA, and then you're going to put that on the PCR and it's going to heat and cool, and it's going to replicate. And then if those microbes are present, you'll see this fluorescence, and you'll be able to detect and quantitate the amount of yeast and mold present. And finally is going to be most probable number methods. And these are culture-based methods, but the principle behind them is slightly different. So you're not physically growing colonies. In this, you're actually looking at specific characteristics. So whether that be turbidity changes or acid production, and you're using that to predict the amount of cells that would grow. So you're not actually culturing it, but it is, in a sense, a culture-based method. So with this, you run all of these different experiments and you get tons of different results, which is the reason for this comparison study. And so first off, what we wanted to do was we wanted to analyze cannabis samples using different types of plates, whether that be agar, petri film, and membrane filters. Plating is a really big controversy, right? Labs, you go to lab A and you get a plating result of this, you go to lab B and you get a plating result drastically different. So why? What is going on there? And so we wanted to be able to compare these methods and assess the reproducibility of the methods both individually and then also against one another. And through this, we were going to also be able to determine how the non-homogeneous nature of cannabis impacts total yeast and mold testing. We all know cannabis is not homogeneous. So what happens if I take a 1 gram scoop here and a 1 gram scoop here? Do I get completely different results? Which result is right? Did I get way more yeast and mold in this scoop and less yeast and mold in this scoop? So that raises questions. And then also to establish true incubation times for each plating method. A lot of the different manufacturers that produce these plates make recommendations. Incubate for 48 to 72 hours. Well, guess what? If you're a cannabis lab and you've been told by a manufacturer you could incubate for a minimum of 48 hours, chances are cannabis labs are just going to proceed with that because turnaround time is important. They need to get those results out, even though that method may need to go longer. And so we wanted to establish that as well. So sample prep and design, what we did here was we took 3 unique samples, and these 3 unique samples were weighed up 5 times, and then we plated those samples at 3 different dilutions. And we used 7 different types of plates, so 4 agars, 2 petri films, and a membrane filter. And then we also took a single aliquot that you see here, and we weighed it 5 times, or we had it weighed once, and then we prepped it 5 times. And the goal of that was to show the reproducibility from in 1 aliquot. So if you have 1 aliquot, theoretically, it should have the same amount of yeast and mold throughout. So we should get the same result for every single one of those samples. So true incubation times, let's talk about that first. So as I mentioned, manufacturers will make a recommendation, 48 to 72 hours, 72 to 96 hours, but that's all they give you. And so it's the laboratory's responsibility to validate that. However, laboratories aren't always the best at that or don't really know how to go about doing that. And so that's what we did here, and these are the true incubation times we established for total yeast and mold for each of these. And the true incubation time was established as the point in time when no new colonies formed, and instead the colonies only started to grow. They just got bigger. So you'll see on the next slide this drastic difference between that 48 and 72 hour, and that's where we're gonna focus, is that Petri film, rapid yeast and mold. This true incubation time is 72 hours, even though the manufacturer recommendations is anywhere from 48 to 72. And that's what you can see here. So you can see here this top set of plates is gonna be the rapid yeast and mold after 48 hours. So with these samples, we put them in. After 24 hours, we pulled them out of the incubator, took pictures. Put them back in for another 24, pulled them out, took pictures. We did that all the way up to 120 hours. And you can see there is a drastic difference between the 48-hour and the 72-hour plate results. And based on this and then reading those plates and attempting to count those plates, I mean, counting the 48-hour plates is not easy. And even when you do count them, you're drastically under-quantifying what that true amount is. We're looking at an average CFU per gram at 48 hours of only 44,000, whereas by 72 hours, we had 165,000. And I did want to note here that, past the 72 hours, that average stayed about the same. We really didn't see an increase, which is why we established that as that true incubation time. Next is going to be the lack of reproducibility we saw between plates. So here is going to be a DRBC and a Sabdex plate, and this is the same exact sample prepped, so it's out of the same tube. We prepped it and plated it on the 2 different plates, and you can see for the DRBC, we got an average of like 127,000 versus an average of 86,000 with Sabdex. Now, if you're in the world of micro, you may say, wait, that's not that different. Those are kind of duplicate numbers. But in the world of Florida, where our regulations are 100,000 CFU per gram, this is the difference between a product passing and a product failing. So if a laboratory was using Sabdex, they would pass this sample. But if a laboratory is using DRBC, they would fail the sample. Additionally is going to be suppressions at low dilution. So this is the Sabdex plates again. And what I want to point out here is that 200x dilution. And so you can see that 200x dilution, that average is only about 25,000 across those. And that's much lower than the average we're seeing for that 1,000x and that 10,000x. And ultimately, I think it's due to oversaturation. There's so many microbes present that no more can grow. However, if you're using this 200x, it is going to drive your overall average down. So it's going to take the average. You can see if you ignore it, your average goes from 61 to 81,000. It goes from 165,000 to 235,000, and from 86,000 to 115,000. So the dilutions you use are crucial if you are plating. You need to be making sure you're using dilutions that make sense for your sample and for your sample type. Then we had the lack of homogeneity and reproducibility. So that sample that I told you, we took the 1 aliquot and we prepped it 5 times out of the same exact tube. These are the results we got. So 110,000, 50,000, 210,000, 90,000, and 80,000. So all over the board. This is, again, the difference between, for states who have a 10,000 CFU per gram limit, this is all failing. It's not a problem. But for states like Florida, where we have 100,000 CFU gram per limit, what do we do here? What is the correct answer? Is this product failing or is this product passing? So conclusions for this portion of the experiment are that, the lack of homogeneity seen in cannabis flower can make it difficult to determine quantitative total yeast and mold results. You may take a scoop here and get one result, and a scoop there and get one result. It may not be due to the method itself, it may be due to the sample. And so how do we create a more homogeneous sample in that sense? Also, the use of different agars or media can lead to variations in the number of organisms that can successfully grow. So certain plates have certain antibiotics, which allow certain organisms to grow, but other organisms can't grow. And so depending on what a lab is using, they may be able to skew those results. And some of them may not even be realizing they're doing that. They may just be using what they've been told is, the gold standard to use for plating, and they do that, and then now they could be under-quantifying those results. Also, the dilutions used during the plating process can impact those final calculations. You need to make sure you're working in the proper range. If you're doing a 100x and your sample has 100,000 CFU per gram, you're not going to be able to grow all of those colonies on a plate, and you're going to skew those results low once you take that average. And then the incubation time for each plate type must be determined prior to processing samples. All laboratories really need to go about this process. They need to take whatever plate type they're using, and you need to let it grow. See what happens. Establish that true incubation time. Truly validate when those samples are ready to come out of the incubator. And then also the use of internal controls is something that I think we should consider. I don't know how to go about doing this, but I figured what better of a conference to kind of say, this would be a great thing to have. Think about qPCR. We use an internal control to make sure our extraction worked, to make sure we did it properly. The chemistry side, we use internal standards to make sure things worked properly. So is there a way to implement something like that into the plating process? With that, we're going to switch to talking about remediation of failing flower products. So with cannabis, if it fails for total yeast and mold or aspergillus, there's a common technique that is to remediate. And whether you're remediating that through lots of different options, but two of the main options are going to be ozone, which is going to be known as, it's a willow is what the most common brand is called, or the rad source, which is going to use x-rays to remediate. So it's going to radiate the product, basically, use the x-rays, kill the yeast and mold, theoretically, and then that will result in the product being safe. We see people doing this, and as a laboratory who runs qPCR primarily, people remediate their product and it still fails. And they're not happy. They said, I killed all of the yeast and mold. And I said, well, I still see the yeast and mold. And they said, well, switch to plating. So we said, we'll look into it. And that is where this study came about. So in this, we wanted to analyze cannabis samples pre and post radiation using qPCR, both qPCR regular and qPCR with Grim Reefer. And Grim Reefer will remove that dead DNA and get rid of whatever they may have killed, and then agar plates and Petri film. We then wanted to add an enrichment step to that plating process of the radiated product to measure cell viability. So one of the thoughts here is sublethal cell injury. The cells aren't actually being killed, they're just being injured. And so if we give them an environment that makes them happy, maybe they'll grow for you, and then you can actually properly quantitate. So we wanted to add this enrichment step to kind of see what happened there. And then also we wanted to look at the pre and post samples for cannabinoids and terpenes to see what that was also doing to the product. So for this experimental design, what we did was we took all of these lovely different samples. So we had pre-rad, post-rad, 2 weeks post-rad, 4 weeks post-rad, 6 weeks post-rad, and 8 weeks post-rad. And I want to mention that the pre and post-rad are the same exact sample. The client literally took a sample, grabbed a handful of it out before radiating it, put it in the radiate, then grabbed more of it and gave it to us. But the 2 weeks, 4 weeks, 6 weeks, and 8 weeks are different samples. They're just those time points out post-radiation. The reason for doing that is to see if, after 8 weeks, have those cells repaired themselves and are now we're going to have tons of yeast and mold growth whenever we go to plate this. So we took all of those samples, we prepped them in triplicate, then we plated them at 3 dilutions using DRBC and 3M rapid yeast and mold, and then we also analyzed those samples via qPCR and qPCR with Grim Reefer. And this is the average CFUs that we saw across the board. So anything in red is going to be those failing CFUs in the state of Florida, so 100,000, over 100,000 CFU per gram. And you can see that that pre-rad sample across the board, it failed. Even with, the use of Grim Reefer brought the number down some, but it still failed. plating, 300,000, 250,000, 330,000, and 142,000. So it's very evident that flower was extremely contaminated, not good. Definitely don't want that flower on the market. The post-rad, however, you can see when we ran qPCR on it, we got 435,000, but then when we plated it, we only saw 1,600 and 1,400. And this is without that enrichment step. So no enrichment step, these are the numbers that we were seeing. So drastically lower numbers across the board. So the theory of the reason why you get higher results with qPCR is because you're not destroying the DNA, but it's dead, seemed to make sense. But we still weren't convinced. We're not 100% convinced that this product is safe for human consumption. And so that's when we added this enrichment step. And so what we did here was, and you can see these little circles that I've circled, these are little dots that are growing. We don't know what they are. We don't know if they're trichome interactions. We don't know if they're just particulate, or are they colonies that are attempting to form, but something has happened during this radiation process, and now these colonies can't grow. I don't know what it is, so I want to figure out what it is. So we took those samples, put them in the incubator for 3 hours, and then we plated them again. And that allowed us to enrich them, give them that time to get excited and get happy, and if they were alive, hopefully to grow for us. And so here is the CFU averages after enrichment. So with no enrichment, for that pre-rad sample, you saw, about 300,000 or so. After enrichment, you saw way, way higher numbers, which tells me that my enrichment process worked. That's why I wanted to enrich that pre-rad sample, is to show that if you enrich it, it's going to grow. If it's there, if it's happy, if it's in the good juice, it's gonna work. And we were able to show that with that. But then post-rad, you can see we did see a slight increase in some of these samples, but nothing drastic, nothing that would lead us to believe that the product was failing, as far as if we're going to plate it. However, that doesn't mean that these cells are not viable, but not culturable. So are they still viable? Are they still dangerous? But can we just not culture them? So looking at cannabinoids and terpenes then is going to be our pre-rad and our post-rad flower here. So to the naked eye, these flower samples may look the same. Also, these pictures may not be the best. However, if you look at these in person and you are looking at cannabis flower all day, every day, like our team is, you get samples in that are radiated, and we can normally tell. We're like, this went through rad source, or this went through willow, because they look almost bleached. They aren't as vibrant. They aren't as green. And so what does that mean? What is that doing to the flower? I don't know. But for a consumer, they may never notice. They may never see the difference in this, and they may never be told that their product went through rad source or went through ozone treatment. So we ran the potency and the terpenes, and you can see that the pre-rad sample, the average potency was 18.3, and then post-rad was 15.4, and then for the total terpenes, we saw 2.37, and then that dropped to 1.98. And so when you calculate the RPDs between those results, they're about 17% for both. So we saw a 17% RPD decrease after radiation. Does this mean this happens to every single sample that goes through the rad source? I'm not sure, but this is what we saw from our preliminary investigation. So conclusions with this is going to be that qPCR and Grim Reefer qPCR yield higher amounts of total yeast and mold in radiated cannabis products than plating. So we're definitely getting a higher concentration. What concentration is right, I'm still not convinced by. Total yeast and mold is a pain. It drives me crazy, but we're here to figure it out, right? The addition of enrichment step can lead to overgrowth in the pre-radiated samples, so you definitely don't want to enrich your pre-radiated samples. However, the post-radiated flower did not yield a drastic increase, but it just makes me feel more comfortable. If there is something alive there, if I give it a chance, maybe it'll grow. So it makes me as a laboratory feel a little bit more confident in releasing plating results. And then any viable cells that may be present in radiated flower do not come culturable for at least 8 weeks after treatment. So that's what we saw there is, we treated they were treated, 8 weeks later, we still saw no growth. Does that mean they're not viable? Again, I don't have the answer to that, but I can say that I was not able to culture them. The cannabinoid and terpene content can decrease after a product has undergone radiation. Whether or not this is an outlier, this is something that happens every time. This could also change based on the amount of time it's in the rad source, the amount of rad source that it experiences, etc. So there's a lot of variables here that in further studies we're going to need to better control. We're going to need to have more information from our client of exactly how long they're being exposed to the x-ray, what rate of x-ray they're being exposed to, so we can kind of better pinpoint, is there a point where you're destroying the yeast and mold but you're not hurting the flower? And then more research is needed to determine whether or not this remediation process is truly removing all of the total yeast and mold, or if it's simply fooling the test. So that's the big takeaway here is remediation able to actually fix our product, or is remediation just figuring out a way to trick the test?
Thanks for your presentation, very informative indeed. As far as I know, one of the health concerns of this microbial contamination is related to the mycotoxins produced by some of those species, particularly Fusarium aspergillus. I understand that remediation might kill those microorganisms, but what happens to those mycotoxins? Are they also eliminated?
So mycotoxins are interesting because they're not a living species anymore like the microbes, so they're secondary metabolites. So with the mycotoxins, when you have a microbial and it starts to break down, you form this secondary metabolite. From our experience, we have seen that even when you extract something, if it previously had that contaminant and it allowed it to form, we've still seen mycotoxins present. So I would say that it doesn't get rid of it. So you're still having a whole nother battle, which is why that mycotoxin testing is so important.
Even if you're able to eliminate the microorganism, mycotoxin still is remaining in the sample. Yeah.
Yes.
Thank you.
I'm sure that they're self-picking these samples, right? The companies are self-picking the sample, right?
Yes.
There's no state or
So
lab that's going.
for final product, we do all of the sampling ourselves. So once the product has been radiated, if it's being sent for final compliance testing, it is already in its package and we go out and randomly select. But for this experiment, they sent the sample. But yes, Florida is very strict about their sampling procedures.
And is there a minimum or maximum to the lot size that you get to pick from? Is it mandated on the volume? You know, is it 10 pounds that you actually pick that sample from? Or is it, 20?
Yes, so it's based on the batch size. So we have to take 0.35% of the batch plus 10 grams for total yeast and mold. So, you're going to take a minimum of 15 grams plus 10 grams. So we're always, as a lab, bringing back a minimum of 25 grams of flour.
25 grams of flour from a maximum volume of?
That would be from a maximum of about 4,000 grams, and then after that, it would increase.
Oh, that's...
So as your batch size increases, once you reach that threshold of the 0.35%, you then have to start collecting more sample.
Well, that's better than
So, there's some samples where we literally bring back like 250 grams of sample.
Oh.
Thank you. Hey, great talk. I have I'm curious about something. When I buy my weed, I don't bring it home and soak it in a broth. So do I need to care about these like residual microbes? I think I would care more about the mycotoxins.
So with the microbes, the thing is that's just allowing us to culture them and to grow them. But standardly, they're still there. That's just allowing us to quantify them by adding them to that broth. But
if they're that hard to culture, then they're probably, it seems like they're probably less competent to infect me too, right?
It's hard to say. It depends on what it is. If you have some powdery mildew, I don't think you want to be inhaling the powdery mildew.
No. I don't want the toxins or the residue from it, right? But when you measure these irradiated samples, I would love to see the error bars and stuff because we do this with so much of our food. Irradiation is brilliant. I've seen lots of data from RadSource and other radiation companies. I think a lot of people suck at using the machines, but
I think RadSource and radiation is used differently in other industries, though. It's not necessarily, it's used for shelf life purposes rather than to kill bad things.
Yeah. Well, and again, right, just the fact that the critters were growing in the weed, that's the bad part, right? And what they were doing, right? They've been crapping in my weed, right? So that's why I don't want it. But yeah, I would find it hard to believe that used properly, this rad source is going to do a significant amount. I mean, I don't do much chemistry with X-rays. That's what we use them for.
And I think that's why more research, ultimately needs to be done before we just put a ton of rad source product on the market, is what is it doing to the product? Make sure it's actually, solving the problem rather than just tricking the test, because there are dangerous molds that could be present. Aspergillus could be present, and rad source could allow that colony not to grow, but the aspergillus then broke down into mycotoxins that now you're inhaling mycotoxins. So there's so many different pieces to the puzzle that I think that the whole picture needs to be addressed.